Hi, my name is Elizabeth Harvey, and I run the UNH Phytolab, short for phytoplankton. And we study all things microbial biology in the ocean. So particularly some things that we're interested in is water quality. So we can enumerate anything from, larger zooplankton down to viruses. We've had a particular experience working with harmful algae and looking at harmful algal blooms. And then we have an interest in human health in the ocean. So understanding pathogens and particularly how potential marine microbes can be vectors of some human pathogens. We use a variety of methodology. So in the lab, we have a flow cam, which takes images of phytoplankton in the water. So we can get some idea of who's there, how many of them are there in terms of species composition and community composition. The really interesting thing is that FlowCam has been used for a lot of different purposes, analyzing microplastics in the water, looking at silica particles or sand particles in the water. A lot of wastewater treatment plants are using a FlowCam to look at how clean or effective their wastewater treatment plant or procedures are working. So there's a wide variety of uses of the FlowCam, all of which are particularly interesting. We also have a flow cytometer, which looks at the very small size classes of phytoplankton. So anything between about 2 and 5 microns in size. So we can count bacteria and viruses on here as well as phytoplankton. We can also use a variety of diagnostic stains to identify particular species if we would like, or some physiological process that they're undergoing. We also have used eDNA to look at changes over time of different marine populations in the water column. And then we also can do pathogenic bacteria enumeration. Here are some examples of results that we have generated from our research. So here's a graph of chlorophyll concentration, both along the outer coast of New Hampshire, but also in Great Bay. So you can see the relative sort of peaks and valleys of chlorophyll. And then using the FlowCam, we can also identify what species are there. So these peaks here are attributed to these species names you see here. These are all diatoms that aren't toxic, but it's really interesting to know in terms of, not all phytoplankton is made the same. So if you're thinking about aquaculture or something like that, the type of species of phytoplankton that are present in the water column make a difference in terms of nutritional content for things that are larger growing in that same water. And also we do look out for harmful algal blooms as well. We can see Studnitia and Alexandrium pretty routinely. We also, here's an example from work that we've done in coastal Georgia, where we've looked at relative abundance of different organisms. In this case, I'm showing the relative abundance of 2 oyster species. So we can use eDNA to track relative population abundance over time. And we can do this for pretty much any species that we're interested in, anywhere from, oysters all the way down to pathogenic bacteria. Please don't hesitate to reach out. I'm really interested. I think particularly our lab could have really nice synergies with people interested in aquaculture applications, anything to do with water quality. And I look forward to hearing from you soon. Thank you. Bye.